Now we're going to start looking into the structure-function relationship for DNA. And so the main functions of DNA are one, stability. So remember that DNA houses, is basically the storage facility for all of the instructions for the cell. It tells the cell exactly how the proteins are made. And so what you want to prevent in any sort of information storage is to prevent information loss. So the DNA, the structure of DNA helps this by being, one, very stable. It's not prone to degradation. And it also allows for several mechanisms within the cell to prevent mistakes happening within the DNA. Okay, so it allows for error checking, for instance, and we'll get to that later. Okay, another thing that's important for this is it can be easily replicated. So remember, part of the central dogma, so DNA, again, is the information storage center. It's the library, right, where you can get instructions in the form of RNA to make proteins. But the other thing that needs to be done is during cell division is that DNA needs to easily be able to replicate itself. Okay, and this is all part of the central dogma. Replication. Okay, and so this has to be easily replicated. And so this is actually facilitated because, let me get rid of this. I don't need this right now. This is facilitated because of the double-stranded DNA. So I'm going to draw a anatomically incorrect DNA. So here's, this doesn't have any of the helix, but we have a 5' to 3' end template strand here and a 3' to 5' end complementary strand. Okay, so you may remember that DNA polymerase and a number of other proteins, so you may remember this from biology. So DNA polymerase takes these strands, and then on this complementary strand, it can be unwound. Right, so that you have the two strands separated from each other. And then DNA polymerase will then complement both of these strands by using, again, Chargas rules, where A will base pair with T, T will base pair with A, G will base pair with C, and vice versa, right? So now, through this, we've replicated, using DNA polymerase, the two strands allow for instructions for creating a new strand. And so with each round of DNA polymerase, then, you get two new strands, okay? So when a cell divides, the information, the genetic information, needs to be passed on to the progeny cells, from the parent cell to the progeny cells. So what needs to happen is you need to have two identical copies of DNA. And so the double-stranded form of DNA allows for this to happen through complementary, because the bases are complementary, okay? So we will look at this a little bit more and now we'll also look at the, I should say, this double-stranded allows for the replication part. Now we'll also look more at the macromolecular structure to see how this facilitates both the replication for DNA and also the stability.